research project this summer is comprised of 3 different portions. The first is further quantifying the data from the 3D cytoprint. The second is visualizing micronuclei within premature aging cell lines using immunofluorescence. And the third is inducing DNA damage within premature aging cell lines in order to see if there is a correlation between the induced damage and a greater presence of micronuclei within the cells. The 3D cytoprint is an application that allows for the A-bulb chambers used for immunofluorescence to be centrifuged. The benefit of this application is that it increases the mitotic cell adherence to the wells, which increases the data that can be abstracted from the experiment. Over the course of the summer, we experimented with different adhesive agents to see if the retention rate of mitotic cells could be increased. We found that the optimal agent that left the wells with the least amount of debris and the most amount of cells was polylysine. In addition to this, we are also working on a padding to coat the bottom of the application to help lessen the force of impact against the wells while in the centrifuge. The second portion of my project is to visualize micronuclei within premature aging cell lines. Micronuclei are chromosomal aberrations that are ejected from the nucleus due to a dysfunction during cell division. Micronuclei form their own separate nuclear membrane, which tends to break down due to its lack of stability. We are able to visualize these fragmentations through immunofluorescence, during which we tag various proteins with antibodies such as RAN, MAB414, lamin B1, TPR, and pericentrin. Over the course of the summer, our goal was to determine the correlation between DNA damage and micronuclei formation in aging cell lines. Etoposide or campamycin CPT was added to the cell lines to induce DNA damage. Etoposide and CPT are anti-cancer drugs that act as topoisomerase inhibitors, causing the DNA strands to break during cell division. To visualize the effects of these drugs, the damaged cell lines were synchronized using narcosol and tagged with various antibodies using immunofluorescence to quantify the formation of micronuclei in comparison to non-damaged cell lines.